I'm pleased to introduce our today's speaker, Dr. Valerie Jones. So she holds a PhD in biochemistry from Louisiana State University, where she focused on enzyme kinetics, and later completed postdoctoral training at Emory University, supported by a prestigious NIH fellowship for her work on host-pathogen interaction. With a strong background in protein biochemistry, enzymology, and experimental pathology, Dr. Jones brings both this scientific expertise and industry insight. So she currently serves as Director of Sales, Marketing, and Technical Support at RayBiotech, where she plays a key role in making complex proteomic technologies accessible and impactful. So we are really thrilled to have her with us today to share knowledge and experience. So with that, I will now hand it over to Dr. Valerie Jones. So thank you once again for joining us, and we hope you will enjoy this session.
Very good. Thank you, Frederick, for the introduction. So let's go ahead and get started. Now, most of RayBiotech's antibody arrays use the sandwich immunoassay principle, so it's really straightforward. You just have a panel of antibodies. Each is immobilized in discrete locations on a planar solid support, like a glass slide or a nitrocellulose membrane. Then you add a sample to the array surface. The target proteins are captured by their antibody at that specific location on the array, and then a cocktail of paired detection antibodies is then added to the array. A signal is generated by either a fluorescent dye or HRP, and then from there, the signals can be detected using chemiluminescence or fluorescence, and so you can obtain a spot pattern. Now, most people will use arrays for collecting protein profiles. That's most commonly used for screening biomarkers or even doing perhaps pathway analysis to figure out cellular mechanisms. But a couple of other things to note about antibody arrays is that they can be quantitative or semi-quantitative. And also that the analyte panels can be small or large. So, for example, small arrays might be a few dozen or a few hundred targets, or they can be large, so-called high-density arrays with multiple thousands of targets. So, for example, 8,000 proteins would be our largest panel. Now, currently, we have 3 different array formats, or should I say solid supports, that are available depending on the researcher's preferences and what equipment they have available. So over here on the left, we have the nitrocellulose membrane arrays, which are easy and fun to handle. And you really just need a standard Western blot imager to be able to run them. Whereas the glass slide arrays here, on the other hand, those can actually be fully quantitative arrays, unlike the membrane arrays, which are exclusively semi-quantitative. And so the glass slide arrays have that advantage. They also have the advantage of having smaller spots. And so because the spots are smaller, that means the overall surface area of the array is smaller too, so they consume less sample. Only 100 microliters of sample is needed per well or per array, compared to 1 milliliter, which is needed for a typical membrane array. So the only thing is you do need a laser scanner to image the slide. But if you don't have access to one, it's not really a problem because Tebubio or RayBiotech USA offers scanning services. So that's convenient because the slides really don't have to be scanned right away. Once you process it, you can actually put it away in storage and scan it later. The fluorescent dye is very, very stable. And then finally, we also offer cytometric bead arrays, which those of you with flow cytometer access will be very comfortable with. But these types of arrays offer exceptional reproducibility and low sample consumption. Only 25 microliters per test is needed. But on the flip side, you're a little bit more limited with the panel size that is available. So the largest panel we offer on the bead-based array is 175 targets. So again, just to recap, you have your planar arrays, okay, the planar arrays are those where the antibodies are gridded or printed onto that two-dimensional flat surface, and then you identify the antibody by its position relative to the other antibodies. And then on the bead-based arrays, this is where each antibody is coupled onto the surface of a microsphere, and then those are identified cytometrically by color. So those are the options we have. Just to let you know, the bead-based array product line is called RayPlex, in case you didn't know. Now, the planar arrays, however, those have a number of different product lines within them. It can be a little bit confusing. We have the quantibody quantitative arrays. We have G series, L series, RayPlex. Sorry, not RayPlex. C series are the membrane arrays. But it's not really important to kind of go over all of that today. I'm just trying to give you a big picture here. And kind of the point I'm making is that we have over 700 different pre-made in-stock arrays in our catalog for you to choose from. So our content is pretty unmatched in the market. And the reason we do that is because we are trying to serve as many different research interests and applications as possible, and as many different price points and budgets as well. So whether you're cytokine profiling, autoantibody profiling, PTM profiling, there's an array for that. So how can antibody arrays help you? Or what kind of research aims can they get you closer to? Well, first off, because antibody arrays are an affinity proteomic tool, it's really the most cost-effective way to profile protein expression in a precise way. So you know exactly what you're detecting right off the bat. Because it's a screening tool, it can keep you from missing something important. It's also a hypothesis generation tool. We see a lot of people using it to sort of determine where to focus next. Also, very importantly, because it uses antibody capture, it is sensitive enough to detect a lot of clinically relevant proteins, particularly cytokines, which are oftentimes present at very low levels in biological fluids. There's also an efficiency aspect there too, in that the arrays, because they work just like an ELISA, they're pretty quick to do. You can generally complete it in one day, the work. And there's also no sample preparation that you really have to do, unless you're working with a tissue biopsy, in which case you have to make an extract. But other than that, any fluid sample you have, you really don't have to do any extraction or depletion before putting it on the array. Really, the only thing you have to do really is dilute it. Okay, so broadly speaking, our arrays can be divided into three different categories depending on their utility. The screening arrays are, over here on the left, are the largest panels that we have. There's just a few arrays in this category, and it's really just our largest arrays that are in that category. So those are the ones that detect multiple thousands of analytes. And those are for open-ended profiling of the proteome. So the targeted arrays are smaller, typically between 10 and 500 antibodies on them. I would consider to be a targeted array. And those can be antibody arrays where the panel is focused on a specific pathway. So they'll be curated based on pathway, molecule type, cellular process, things like that. So those are kind of more for hypothesis-driven applications. And then third, we have the post-translational modification arrays, which are a specialty type that are actually a screening array also. So these arrays can be very large, but they have a pretty specific application. So for example, if you're interested in protein acetylation or protein oxidation, there are arrays that can do that. Now, I kind of alluded to this before, but arrays are a mature technology at this point, and they've been extensively cited in the literature. So we have seen them be used in multiple different applications, including assessing cancer drug action, biomarker discovery, clinical trials. And some of these approaches I will actually give specific examples of today. Okay, now with the product education out of the way, let's go over a couple of case studies. And specifically, the ones I'm going to show here are immunotherapy related. So the question we want to explore today is, how do we distinguish immune checkpoint therapy responding patients from those patients that don't respond? And so the way that many researchers have been approaching this is to start by characterizing the host anti-tumor response. And we'll get into that. But this figure here illustrates how immune checkpoint inhibitors can wake up T cells by targeting these immune checkpoint receptors, specifically PD-L1 and or CTLA-4. Now, how well this works is going to be largely shaped by the cytokines of the tumor microenvironment, which can either promote immune activation or create an immunosuppressive environment that can get in the way of the drug working. So if you look at the cytokine profile, you might be in a better position to look at therapy response and maybe even figure out the mechanism of resistance. But there's a problem, which is that what markers do you look for and where? Okay, so first off, the current clinical practice is to look at tissue-based tumor biomarkers to decide if the patient is a candidate for immune checkpoint therapy or not. So those might include PD-L1 expression in tissue, so that's measured histologically. Could be tumor mutational burden, or TMB. So that just refers to the number of mutations in the DNA. Microsatellite instability, and a few other things like that. But those methods are all biomarkers that require a tissue sample. So these types of biomarkers are a little controversial, and part of the reason is that biopsies can give an incomplete picture of the tumor. They're also invasive and hard to obtain. And actually, because it requires surgery, the patient's condition might complicate that process, and that could delay treatment decisions. And then another thing is that these tissue biomarkers are also impractical for longitudinal studies where you have to take multiple samples over time. So that's another issue. So for that reason, peripheral blood biomarkers are increasingly sought after because they do allow for that serial monitoring over the course of a treatment, and also they're a lot less invasive and costly to obtain. So I'd like to show you a study here that focuses on non-small cell lung cancer. And this group wanted to address the low success rate. Okay, so only 20 to 50% that is observed for patients undergoing immune checkpoint therapy for lung cancer. So they went after a predictive signature from plasma. And their approach was a machine learning algorithm that was trained on both clinical data and proteomic data from plasma. They obtained the proteomic data from a RayBiotech kiloplex array, which is a 1,000 target quantitative array. And the proteomes were collected longitudinally over 143 patients receiving anti-PD-1, PD-L1. And just a quick note here, T0 refers to the pre-treatment data point, and T1 refers to the on-treatment data point. So just for reference on the next slide. So with that approach, they did indeed identify a predictive signature at T1, consisting of two proteins, okay, CXCL10 and CXCL8, as well as sex and age. So 4 parameters. You have 2 proteins and 2 clinical features. And just a quick note here, those 2 predictive proteins were present at significantly higher levels in the non-responders compared to the responders at both time points, T0 and T1. Also a quick note, CXCL8, which is IL-8, is secreted primarily by neutrophils. So anyway, they found that these biomarkers were indeed predictive for treatment response. They also identified 3 biologically and clinically distinct patient clusters from the data. And these plots right here are Voronoi plots, and they show the functional groups of proteins that they found present at higher levels at T1. And so what they found was that neutrophil-related proteins are enriched in cluster 1, which is the non-responder group largely. And that also there were higher levels of various different metabolism-related proteins, so including pyrimidine, purine, and glycan metabolism proteins were enriched here, and some of those are associated with immune suppression. So kind of having this better understanding of the biology of these non-responsive patients is useful because it can reveal new drug combinations that can be tailored to that patient to block the pathways associated with resistance that they found here. Okay, so this next study is again about lung cancer, but this time this group is focused on stage 3 lung cancer. So here, the problem being addressed is that tissue-based biomarkers, like I mentioned, the PD-L1 in tissue, TMB, they're not terribly useful in clinical practice for anti-PD-1 therapy because, like I mentioned, just they don't have a high success rate. And furthermore, this group, because they were looking at stage 3 patients, they actually are investigating neoadjuvant therapy. So that means combining 2 different treatments, in this case, chemotherapy and immunotherapy together. And so tissue biomarkers is really even less useful in this regard. And so the background here is that they had data from a previous clinical study with stage 3 patients showing that when you combine nivolumab, which is anti-PD-1, and a platinum-based chemotherapy, it gave 63% complete pathological response. So pretty encouraging. 63 is not bad. And complete pathological response means 0 tumor cells, okay? Neoadjuvant therapy did look pretty promising in this clinical trial, but they wanted to be able to identify these complete responders beforehand. And so in this study, they had 46 stage 3 patients. They collected plasma and PBMCs pre and post treatment. And then plasma proteomics was done using the G-series cytokine array 4000, which is a protein array, an antibody array that detects 200 proteins semi-quantitatively. And then they also did immunophenotyping on the PBMCs. So baseline profiling showed that the completely responsive patient had lower levels of neurotrophins, specifically NT3 and beta NGF, also had lower levels of VEGF-D compared to the non-responsive patients, and then also lower levels of inhibitory cells. So that kind of suggests overall there was a less suppressive immune microenvironment. Now that was at baseline. So then after treatment, what happened was the responsive patients had increases in immune activating markers. So MCSF, PARC, decreased levels of immunosuppressive factors like MPIF1. So that suggests an enhanced anti-tumor response, perhaps by immune reprogramming. So, this was an exploratory study, really wasn't powered enough to detect biomarkers. But again, it points to the distinct biologies between the complete responders and the non-complete responders pre-treatment using parameters that are measurable in peripheral blood. So it's a really great proof of concept. Now, this next study is a preclinical study, and it was focusing on treatment resistance in triple-negative breast cancer and treatment-resistant melanoma. So the rationale here is that patients who relapse after immune checkpoint therapy simply fail to generate a lasting adaptive immune response to the tumor. And it's thought that this might be due to deficiencies in tumor antigen presentation. So they asked the question, is it possible that if you activate innate immunity, that you could trigger a better immune response, strong enough to overcome resistance to that immunotherapy? So just a quick recap here, innate immunity is the first line of defense against pathogens, and toll-like receptors or TLRs play a central role in that. So TLRs can directly recognize bacterial and viral products that come into contact with the cells and trigger an inflammatory response. So this group chose to activate the innate immune system using a TLR5 agonist, aka bacterial flagellin, or alternatively, the peptide CBLB502, which is an analog. So the idea here is that they would activate the innate immune system to shift the tumor microenvironment out of that immune suppressive state and hopefully bring about a lasting anti-tumor response. There's actually a lot of precedence for this. You might be well aware, but plenty of papers have attempted this either through TLR5 agonists or other TLR agonists. But anyway, in this study, their strategy was to use a mammary carcinoma mouse model. So they implanted tumors in 4 groups of mice, either treated with immune checkpoint therapy alone, so that would be anti-PD-1 and anti-CTLA-4, or flagellin alone, or both drugs in combination, and then they had their vehicle control. So to look at the soluble factors in the mouse serum, they used a mouse-specific 32-plex array. And they also looked at tumor infiltrates. So they isolated cells from biopsy tissue. And during this study, the only mice that survived mainly was a portion of the mice that received the combination therapy. So 20 to 25% survival in that group. And by the way, they showed that that survival was TLR5 dependent. They used a knockout mouse to confirm that. So the next step was, out of the mice that survived, they performed a tumor re-challenge. So they reimplanted tumor cells again. And surprisingly, they observed an 80% survival rate after the re-challenge. So that kind of shows that the surviving mice had acquired memory for anti-tumor immunity and simply rejected the tumors. So the cytokine array data and cell-based analysis together showed that this combined treatment causes upregulation of IL-15, which is an immune activating cytokine, as well as upregulation of CD3 and CD4 positive T cells, and a reduction in GCSF and CXCL5, which are both immune suppressive cytokines, and a reduction in monocytes. So taken together, they hypothesized that this combination treatment can shift the tumor microenvironment from immune suppressive to immune activated through those changes. So, this is another case where, using the TLR5 agonist, flagellin or CBLB502, in combination with immune checkpoint therapy, could be useful and could provide a new opportunity for these previously unresponsive patients with these difficult cancers. Now, the publications that I've snapshotted here, but I can't really go over in detail today due to time, are a little bit different. These are mostly biomarker studies where the RayBiotech array was used for cancer diagnosis or prognosis for the most part. Although this publication here on the top left is another immunotherapy response prediction study. It's actually a very recent one. But anyway, these are just a few examples where the RayBiotech array was used to find helpful signatures. And then here, these studies were more mechanistic in nature. So they focused more on mechanisms of metastasis or mechanisms of therapeutic efficacy, why the drug works, or even mechanisms of targeted drug resistance, which targeted drug resistance is a whole other topic that could fill another webinar, but there is a large body of research on that too. Now, I did want to present one more use case because since we're still on the topic of the tumor microenvironment, but here, instead of looking at plasma or serum for answers, this group, which was out of Harvard and Broad Institute, looked at stromal cells, specifically cancer-associated fibroblasts. And this group speculated that CAFs, cancer-associated fibroblasts, may have molecular features that could reveal some insights about personalized treatment strategy. And the reasoning is because there's such a high degree of heterogeneity in calf phenotypes. So they wanted to take a closer look at this, and their approach was to capture the diversity of these calves in non-small cell lung cancer and study their functions. So they derived a large collection, a large biobank of patient-derived fibroblast cultures, or PDFs, from biopsies that had targetable oncogenes. So specifically, they looked at EGFR or ALK mutations. So in other words, cells that had targetable genes for tyrosine kinase inhibition therapy. So they had 80% success rate with deriving those PDFs. And then using a combination of RNA-seq, immunophenotyping, and secretome profiling, which for that they used a customized quantitative proteomic array from RayBiotech. They identified 3 functional subtypes, 3, that magic number coming up again, of CAFs. And long story short, those CAF subtypes are defined by their expression of HGF and FGF7. So subtype 1, CAFs have high expression of HGF and FGF7. Subtype 2 have only high expression of FGF7. And subtype 3 has neither highly expressed. But even more importantly than that, they demonstrated a link between patient clinical response to the tyrosine kinase inhibitor and the functional classification of the calf that was derived from that patient. So they found that that first subtype, the one that highly expresses HGF and FGF7, was robustly protective of cancers. The second subtype, the one highly expressing FGF7 only, was moderately protective, and the third subtype provided minimal protection of cancers from the drug. So this was pretty cool because these 3 CAF subtypes actually pointed to a personalized treatment strategy to overcome this calf-mediated therapy resistance. And so specifically, they proposed that if you block the MET plus FGFR pathway together, that could be useful for combating the impact of those subtype 1 CAFs, or FGFR pathway blockade alone might be needed to block the influence of subtype 2 CAFs, and then no combination therapy would be needed for subtype 3. So that's all I wanted to cover today. Just one final comment, which is that if you're interested in one of these large high density arrays, the ones that have over 1,000 targets or more, you may want to consider doing a service with Tebubio's team just to ensure that you get the most from your investment. Their team of experts is highly qualified, and we trust them very much. So, Frederick, if you want to take control again.
Thanks a lot, Valerie, for this very clear and precise presentation. So before moving to the live Q&A session, I really would like to emphasize that the technological advances made by RayBiotech in their multiplex proteomic analysis tools combine, as mentioned by Valerie, with our laboratories collaboration to offer biomarker mapping services in Europe, or really accelerating the understanding of the tumor microenvironment and the development of the new therapeutic approach. But okay, now it's time to ask all your questions and gain insight from directly from Dr. Valerie. So please use the Q&A box at the bottom of the of your screen, and I guess we have more than 10 minutes to ask all your questions. So please feel free. Okay. Let's move to the first question. First question, what are the specifications to generate a custom array, minimum number of targets, minimum number of slides, and what is the delivery time?
Yeah, so I did make a reference to a custom array within this slide presentation. So good question. So the minimums and the specifications depend on what type of array you're ordering. So just to give you a quick idea here, for example, if you want a membrane-based array, we have a minimum of 10 membranes and 5 targets. If you want a RayPlex array, so one of those bead arrays, the minimum order is just 1 plate. So 196-well plate, which would be 80 tests. And the smallest array we can make for RayPlex is 2 targets. If you want one of these larger arrays, for example, the platform that can do 8,000, but you don't have to make it that big. The minimum order for custom would be 20 arrays, and minimum number of targets would be 5. And then for the quantitative array, we have a minimum order of 16 arrays, and then 5 targets is the minimum. So it's a little bit different depending on the platform. But if you do place an order for custom array, it can take around 2 to 3 weeks typically, unless it's a very large order, in which case we might need a little bit longer because you need more array printing time. And generally, the cost is about 25 to 30% more than a comparably sized pre-made array. So just to give you an idea what you're looking at there.
Thank you, Valerie, for this precise answer. We have a second question from an attendee. When should the customer, the researcher, have to choose quantitative versus semi-quantitative array?
Yeah, quantitative versus semi-quantitative. We talk to customers about this a lot, and a lot of it just comes down to the researcher's preference. But if you want some guidelines, one factor to consider is just the cost. Quantitative arrays are going to be a little bit more expensive per target, usually compared to a semi-quantitative array. And the reason for that is because there's an external calibrator. You have to make a standard curve for every target. So that adds to the cost. But that extra cost can be worth it for a researcher if they are looking for that higher degree of confidence and precision. Because you are comparing your results to an external calibrator, it is a little bit more accurate, and you have a more defined confidence interval. So it's more clearly defined where the signal becomes background versus when it's out of the range of linearity. So that can be really appealing to people. And then also quantitative can be important for if you're testing batches of biological samples over time and you want to compare across, or if you want to establish some clinically relevant levels and ranges that could be compared across studies, then usually people are going to prefer quantitative arrays. Now, semi-quantitative arrays do have their place too, because especially if people are doing experiments testing cell culture media or testing cell lysates or even tissue lysates, a lot of times semi-quantitative arrays are just as informative as a quantitative array. You can get the information you need as a relative fold change in signal, and that can get you what you need to know. Also, semi-quantitative arrays are great for doing screens. So particularly if you're doing one of these large arrays, these high density arrays, it's really useful for just detecting which biomarkers are important to start off with, and you save a bit of money by doing that too. So that's what I would say to consider when you're trying to choose quantitative versus semi-quantitative.
Thank you, Valerie. Two new questions came. So the first one, how sensitive is the method? How low protein concentration is needed for samples, and how much sample is needed?
Oh, right. Yeah. So the sample concentration. Okay. Now, normally we say that if you have a biological fluid like serum, for example, or even a different biological fluid, let's say you're looking at cerebrospinal fluid or something like that, you are normally able to detect cytokines with any of the arrays that I talked about here most of the time. And we normally suggest doing like at least a twofold dilution. The twofold is usually good in most cases. Now, if you're having a sample type where you have to do an extraction, like you have tissue, we would say dilute it a little bit more, because tissues can be a bit concentrated and you do have to dilute out a bit of that lysis buffer before coming into contact with the array. So when you make a tissue extract, you're generally looking at, we say try to get 10 milligrams of tissue to start off with. That would be a good amount to create your extract. And then you want to bring it up in, let's say, maybe 500 microliters of lysis buffer. You can use a little bit more. And then you want to measure the total protein content of that. And the total protein content you want to look at will be somewhere between 50 and 500 micrograms per mil of total protein is what you want loaded on the array. So your actual lysis concentration will probably be more concentrated than that. So you will have to dilute to reach 50 to 500 microliters of material. Okay, so like I said, that goes for cell and tissue extracts. But when you're talking about just body fluids that you extract directly from your animal or your patient, there isn't much you have to do there. If you're using a quantitative array, you're generally going to need 50 microliters per well. That's assuming you're going to do that twofold dilution. Okay, so you need that 100 microliter working volume. If you're using a membrane-based array, you're going to need more sample. You're going to need 1 mil or 500 microliters. And so usually people are using the membrane arrays for doing like cell culture experiments. So pretty ideal for stuff like that. But we've seen lots of people use serum too. And then for so if you're really limited on sample and you can't afford to spare 100 microliters of something, then you may want to look at RayPlex because that is the least sample consuming platform we have. So that one is 25 microliters per array or per well is what you would need there. And then for sensitivity wise, I believe you asked about that too. Almost all the arrays can detect picogram levels of proteins most of the time, somewhere between picogram and nanogram. It depends on the antibody. So that is sensitive enough to detect cytokines in most cases. And so, yeah, hopefully that helps.
Thank you, Valerie. Next question. In different type of assays, are there any differences in the requirement for sample preparation?
Yes. Not really. The only thing I can think of that is a difference would be the large high density arrays that are label based. So that really large 8,000 plex. So we have an 8,000 plex and a 6,000 plex, and those actually have a labeling step. So that is one extra step you have to do. We actually require to biotin label all the proteins in the sample before loading them on the array. Reason being, it works by antibody capture, but the detection, you detect the protein directly using that biotin tag. So that biotinylation step is an extra maybe hour of work. It doesn't really add too much to your workflow. But that's really the only difference. Each array does come with its own diluent buffer that might be slightly differently formulated, but the way that you use it is essentially the same.
Thank you, Valerie. Next question. What is the dynamic range, rough measure, for detecting most proteins?
Yeah, so I'll start with the quantitative arrays. So that would be the quantibody array and the RayPlex arrays. You're typically looking at 4 orders of magnitude. It can vary a bit. It depends on the antibody. Some antibodies have a really nice range of detection, and some antibodies are a little bit shallower. But 4 orders of magnitude is typical. And so you're talking like low picograms to, nanogram levels easily. And then for the C-series membrane-based arrays, it is a little bit smaller. So typically that signal you're looking at is going to be anywhere from like 0 to like maybe 6,000. So we're talking 3 orders of magnitude for that. Still pretty good. So yeah, whereas the glass slide arrays have a larger dynamic range, you're typically going from 0 to 60,000 or maybe even 100,000. So that's your signal range. That won't necessarily be your linear range, but it will be the range of detection of a signal.
Perfect, thank you. There is maybe a second part of this question. Also, in a quantitative assay, what is the limit of quantification? Picogram, nanogram?
Yeah, so that's a good question, and it does depend on the antibody. So it's different for each target, you know. We do have those specifications listed in the manual of each of the arrays. That they are all tested, the LODs. But typically, like let's say for cytokines, it's not uncommon to see single digit picogram, but sometimes you get single digit nanogram occasionally, like I think for TGF beta, it's a little bit higher, but TGF beta is normally present at higher levels anyway. So yeah, it can vary quite a lot, but yeah, but these like low to double digit picogram per mil levels are not uncommon for a lot of targets.
Thank you, Valerie. Maybe we still have time for a few questions, so don't hesitate to ask directly to our experts. I have another question. How do the throughput and reproducibility of affinity arrays compare to mass spectrometry-based proteomics?
Yeah, that's a good question. So, mass spec and antibody arrays, actually, the reproducibility is kind of similar. Um, they're both they can run under 20%, broadly speaking. And so that's, of course, you have to realize like that the factors that contribute to variability in those two platforms are completely different. And so this is kind of like assuming that you take every precaution to minimize every variable that you can. That yeah, you can achieve under 20%. And so intra and inter CVs for arrays is usually like it's usually 15 to 10 to 15% is not uncommon. Um, and then actually, like I mentioned before, the Rayplex beta arrays are even better. Like those actually are the most, high reproducibility platform, and those are like hovering around like under 5% usually for inter and intra assay CV.